Hello, everyone. Thank you so much for joining us today. I'm Sean Buchanan, Assistant Director of Development for the Physical Sciences Division here at the University of Chicago. I want to welcome you to today's special presentation on the topic of learning physics of living systems, in which we will hear from some of our outstanding faculty on their pioneering research in this field. Before we get started, we'd like to ask that you remain on mute when not participating in discussion or asking questions of our presenters. We have allotted time after the presentation for Q&A discussion, but we also encourage you to share comments or questions during the presentation itself by making use of the chat or utilizing the raise hand feature so that we can hear from you directly. Our presenters have also designated times during their presentation to solicit input from you directly. Alternatively, if you wish to have your questions or comments remain anonymous, please send them directly via chat to myself or my colleague, Amy Keating. I'd also like to note that this meeting is being recorded so that we can share it back with all of you after the discussion. So please expect a link and a follow-up email. With that, I'd like to pass it over to Young Ki Kim, Lewis Block Distinguished Service Professor and Chair of the Department of Physics, who will give some brief remarks and introduce our speakers.
Thank you, Sean. And hi, everyone. Today, my physics colleagues, Margaret Gardel and Vincenzo Vitelli, will discuss recent progress on physical principles of living systems, spanning from microscopic collections of biological molecules to bird flocks and robot swarms. As Sean mentioned, we'll start with the short presentations, about 50 minutes each by Margaret and Vincenzo. They will give a gentle introduction to recent experimental progress, theoretical concepts and approaches based on artificial intelligence that have emerged at the interface between matter, life, and machines. Now introducing Vincenzo Vitelli and Margaret Gardel. This is the order of their presentations. Vincenzo is a professor of physics and a member of the James Franck Institute at the University of Chicago since 2017. Previously, he was a professor in the Lawrence Institute for Theoretical Physics at Leiden University, where he held the chair of condensed matter theory between 2015 and 2017. He was an invited professor at PCI as a Juliette Curie Chair in 2013 and Economel Paris in 2015. Vincenzo obtained a PhD from Harvard University in 2006. He was a postdoctoral fellow at University of Pennsylvania. His honors and awards include a fellow of the American Physical Society, the H. Callan Prize, the Feinberg Foundation Fellowship from the Weizmann Institute. Now, Margaret Gardel. She's the Horace B. Horton Professor of Physics and Molecular Engineering, Director of the James Franck Institute at the University of Chicago, and she's a member of the Institute for Biophysical Dynamics. Margaret joined the University of Chicago in 2007 after earning her PhD from Harvard University and completing postdoctoral research as a Pappalardo Fellow at MIT and at Scripps Research Institute. Her honors and awards include a Fellow of the American Physical Society, the Packard Fellowship, the Sloan Fellowship, NIH Director's Pioneer Award, and the Raymond and Beverly Secular International Prize in Biophysics. Please welcome Vincenzo Vitelli and Margaret Gardel.
Hi, everybody. My name is Vincenzo, and I'm going to be starting this presentation. Let me share screen. There we go. One second. Hopefully you can see the screen. Yeah, perfect. All right, so I would like to take you on a journey that will go from robots, living system, and machine learning. And it's a special pleasure to do that with Margaret. It turns out that Margaret and I know each other for a while. I mean, we went to grad school together, but we haven't worked much together until very recently, a couple of years ago, when we started this collaboration that really brings two things that, as you can see, are quite different from each other, and it's been a real fun. So the presentation will reflect that. We'll swap back and forth from each, with our presentations. And I would like to encourage to just interrupt. If you have a question, comment, objection, it's much more fun to do so in this interactive way. My PhD advisor used to say it's better to uncover a little bit than to cover too much. So in that spirit, please interrupt our presentation. I will just answer your questions. So what you're going to see here already on the screen is a bunch of moving objects. On the left is a bunch of robots, and on the right is a cell crawling. And I would like to document the journey that took the two of us through connections between this type of systems. But before that, I'd like to start with a very, very simplistic view of what physics is. I mean, it's a monotheistic subject. Most of physics concerns minimizing energy. So in many contexts in physics, no matter how complicated the physics you're looking at, what you're doing is you're viewing the system a bit like this ball that goes down in an energy potential, and by the time it goes at the bottom of that potential, it sits there. The assumption is that there is a state called equilibrium where the system is happy. And the dynamics of the system, the time evolution, it's just a motion towards that special state. Okay. I'd like to convince you, though, that there are many systems in nature at the interface between physics, biology, and engineering that really do not fit this paradigm. In one case that I'm going to start with, it's a case in which the fundamental building blocks of the system, represented here, for example, by this red metronome or this blue metronome, interact in non-mutual ways. What do I mean by non-mutual ways? Imagine a system in which A wants to synchronize with B, but B does not want to synchronize with A. And you have many such components in your system. Well, you can see right away from the perspective of the microscopic interaction, there is a frustration. These two things do not want to do the same things. Not surprisingly, perhaps, because of this microscopic frustration, the system cannot simply reach a state in which it's happy, in which it can stop moving. So it cannot be described using this paradigm of a ball that relaxes in the minimum potential. Instead, it's condemned to move forever. And that motion is what we want to understand, that perpetual motion. Now, this is not such an unusual thing. For example, if you can see on my screen here, an example of non-mutual interaction outside of physics in ecology is the relationship between predator and prey. It's pretty obvious that this rabbit interacts with a carrot in a very different way than the carrot interacts with a rabbit. And in fact, even in our own brains, the interaction between neurons entails this non-mutual interaction. In the brain, we have inhibitory and excitatory neurons. And as the term designates, they do different things on each other. So studying this system with non-reciprocity, it's the rule rather than exception in nature. But starting from elementary mechanics, we've been taught otherwise. One of the first things I learned in elementary mechanics was Newton's third law, namely the force of A on B is the same as the force on B on A. So what I would like to do today for you is to start from elementary mechanics and show you some systems that break Newton's third law. And so what you're going to see here on my screen is a simple device built in collaboration with Corentin Coulet in Amsterdam, who sits here with his robotic platform. And I would like to show you what happens on this innocuous-looking system of strats when you touch them. In particular, I will first touch the left rod and then the right rod. And I invite you to monitor what happens to either of them. So let me get started. So here, when I push the left rod, the right one comes in. So let me do it again. I push in, the right one comes in, as documented in this image right here. Now I want to show you what happens if I touch instead the right rod, okay? When I touch the right rod, the left one goes out. You see, now if I push in with this one goes out. It's exactly the opposite that happens when I touch the left one. So there is a basic asymmetry in the system in the way it responds. If I touch on the left, the right one comes in. If I touch on the right, the left one goes out. Now, this system can behave in this way that apparently breaks down Newton's third law, because it's not a simple system of passive strats. These strats are connected to controllers and motors, which have internal sources of energy that can be leaked in the system. They have electronic feedbacks. And now, because of this constant injection of energy via the motors, this system mechanically behaves in very peculiar manners. And one of the most important features that it has, since the forces that which is built cannot be expressed as a gradient over potential, when you bring the system around the loop, it can generate work. And this is particularly obvious if I build a ring out of those building blocks that you saw, and I just touch it. If I do that, what happens is the following. What you should be seeing on your screen is this ring to continuously move, undergoing what appears a cycle of deformation. The peculiarity of this cycle is that if you don't touch the system, it will stay there. But once you touch it, it continuously moves. It simply cannot stop because of those non-reciprocal interactions. Notice this cannot be described as something that stops at an energy minimum because it moves forever. Now, when we saw this, we thought, okay, can we actually use this for something? Can we build a machine out of it? So what we decided to do is to, if you like, this is a bit of a cheeky title, I admit, the rediscovery of the will. We tried to think again at this perhaps most basic of human invention, the will. So you all know how the wheel works. I mean, you know, you have a car. I mean, I don't have a car, I have a bike, but it's an electric one. So the principle is the same. The wheel is connected to a shaft. The shaft rotates and the wheel moves. Now, this wheel is special. This wheel will move when you touch it, when you deform. And rather than using many words to describe it, let me just play a movie. Here is what happens. I don't know if you can see, as this wheel bounces up, it deforms. And the more it deforms, the more it undergoes those cycles. And the more it undergoes those cycles, the more energy it generates from the motors. So this is a wheel that moves because it adapts to a certain terrain. And in particular, if you take this wheel that moves on a rough terrain, it will adjust the speed at which it moves to the terrain, because the more it bounces against the asperity of the terrain, the more it deforms and the more it moves. It's an adaptive object. It's a machine that can respond to its environment. Now, this object, and you can read it a bit more if you want, we call it the odd wheel in this article in Quantum Magazine, which is a popular journal from the Simon Foundation. It's an example of an engineered structure using building block that generates energy, but is by no means the only things in nature that has this property. There is a different set of machines that function, that are not created by us in laboratories, but they're biological machines that arises from biochemical processes. And what I'm going to play here in the background is a cell crawling. This cell moving, like the odd wheel, harnesses energy generation at the microscopic level, but the mechanism behind are remarkably more complex. And what we've done in our partnership with Margaret is to try to understand, using physics principle and something more, how this works, how this mechanism of propulsion and locomotion happen also in biological systems. And so in the spirit of this talk, I'm going to pass the ball to Margaret now to tell you a little bit more about this biology, and then take back the task of explaining some of the mathematical models and machine learning model that we're doing to understand. And this would be a good time to ask some questions if you want on the first part, otherwise there will be plenty of opportunities.
Should I transfer over? Are there any questions?
Yeah, looks like there are no questions at this moment. So Margaret, go ahead.
Well, great. Can you just unshare your screen, Vincenzo?
Yep, done.
Great. Perfect. Okay, thank you very much. My lab has been interested in understanding what controls motion and shape in biology, right? And so we're very familiar with this at the macroscopic scale of that. As we develop from a young child, you know, our organs have to grow to be the right size and proper size throughout development, and our muscles have to learn how to coordinate motion at these macroscopic scales. Now, of course, when the light microscope was invented by Galileo, you know, several hundred years ago, this allowed us to see these types of motions and try to understand what the microscopic origins of these motions are within living systems, right? And so these are three different types of movies that to a certain extent could all have been seen several hundred years ago, right? So in the early 1800s, right, Tom Brown discovered Brownian motion, the leaping of pollen grains within an aqueous fluid, right? And then you can also see the motion at a little larger scales of individual cells. This motion of cells is essential for healing wounds in our body, building new vasculature, to build blood vessels, and also to support our own development. So on the right hand is a series of time-lapse image of the first 24 hours of zebrafish development. So all these types of motions, right, we want to understand the physical principles about how biological or living matter can really support them at these microscopic scales. And of course, the one that we know very well is Brownian motion, right? With Einstein coming up with a theory, understanding how thermal forces can really drive random kicks on these microscopic particles to drive their random motion. The questions of how cells crawl and how cells control their shape and development is still one under active research. It's been one, of high interest to scientists over the past 100 years, and starting from Sir Darcy Thompson, who wrote this wonderful book on growth and form, where he started to consider what types of simple physics might be at play in understanding the deformation and forces at play in these types of soft materials. And one analogy he used quite often was this idea of a collection of compressed soap bubbles. So if you take a lot of soap bubbles and compress them in shape, they distort their shape, and their shapes are all determined by surface interfacial interactions between the soap bubbles. Now, so even though this has been of interest of understanding how these soft biological matter control their shape, what was really missing is a the equivalent of an atomic theory of biological matter. And that's really where this field got stuck. And over the past 100 years, what modern biology has allowed us to do is to reveal the molecular basis of life and living systems. So 20th century biology has really been being played out at this molecular scale of understanding the structure, function of biological molecules, and how they can build different systems inside cells. What is really fascinating to me is that this 4 orders of magnitude difference in length scale of how do you take these collections of molecules to build materials and machines that then support locomotion and shape control at the cellular and tissue scale. But we really physicists required this molecular discovery in biology to be able to enable further progress. So Vincenzo already showed one of these movies, but really the type of data that we collect today in this field really relies on recent technology developments over the past 20 years or so that have allowed us to visualize protein dynamics inside living organisms and visualize these as really high resolution using modern light microscopy. So there are now fluorescent proteins that get excited by when illuminated with a certain wavelength of light and then emit at a slightly lower wavelength. You can barcode different proteins by tagging them with different types of fluorescent proteins. And then using modern tools of gene editing and gene expression, you can express these fluorescent constructs inside your cell where they're tagged to certain locations. And then using modern light microscopy, you can visualize these with very high resolution to get information at the subcellular scale of these protein dynamics over time. And this allows us to get very quantitative measurements of protein dynamics within living organisms as they're moving or developing. And what this looks like in my laboratory is a confocal microscopy. I'm not going to go into the details of everything here, but we put our sample at the top of this inverted microscope. There's a lot of lasers for multispectral imaging. And then the confocal microscope is a way to sculpt the light beam to control precisely where you're illuminating your sample to eliminate all your out of focus light, and then only collect the light within your focal plane. So these tools, we have several of these in the lab so that we can do many different types of experiments. On this particular instrument, what will come up later is another laser line that allows for, through a different port, for parallel activation of light in addition to illumination for further control. So, when we look inside cells and ask, what the physics of their material of their inside is, we can take these really pristine images of the structures inside cells. There's these biopolymer networks that control the scaffold and the mechanical structures within them. This is the biopolymer of actin filaments shown in an individual cell that's migrating or in multicellular tissue. And again, over the past 30 years or so, there's been a lot of progress in understanding the machines, the macromolecular machines that generate protrusive forces at the front edge of cells to drive its forward advance, the machinery that controls where the cells are sticking to in terms of their adhesion to each other or the surrounding environment, and also how they act as tiny muscles. They contract against an environment to generate contractile forces. So all these machines now can be characterized and understand the mechanics and physics underlying these macromolecular machines. So that's what we've been spending a lot of time doing, is understanding what the physical origins of these sort of large disordered collections of macromolecular machines. As I mentioned, a lot of a whole field of sort of single molecule biophysics has characterized individual molecular motor function. And so the machinery that's involved in contractility is the molecular motor myosin 2 that then walks and binds, and then through the hydrolysis of a chemical undergoes a conformational change that then drives the translocation of that biopolymer actin. So just like a car motor, this is now a molecular scale motor that has a stall force. That's the force at which the motor stops translocating. And then it has a maximum speed when it's under zero force. So these motors then drive rapid displacements of the biopolymers throughout the system. But what we were interested in is understanding how these molecules self-organize to build contraction at the cellular scale. So I made the analogy to muscles, and striated muscle is where these have been found most ubiquitously. And so here in striated, in your voluntary muscle of your skeletal muscle and your cardiac muscle, the way the system contracts is because of its organization into sarcomeric organization. And what that means is that these molecular motors are organized so that they're at the filaments with the right polarity. So when they engage and translocate the fibers, the whole local unit gets shorter. And then these sarcomeric units are linked in series across the cell to drive contraction at the cellular and even tissue scale. So everything in this system is highly geometrically regulated, although I will say that how this system self-organizes in striated muscle is still not well understood. But the mechanics is pretty simple. It's a geometrically regulated contraction that scales molecular scale motility up to larger scale contraction. So, so we were interested in understanding how the same types of molecules, this actin and myosin filaments, can drive contraction in systems that are very highly disordered. So these are the same molecules, actin and the molecule myosin 2 in a single cell and in multicellular tissue. And in most tissues in our body, this is what the systems look like. You don't see any of this sarcomeric organization. It's highly disorganized. And so we wanted a way to be able to understand how contractility still arose in these systems. So what we did was we take these proteins, isolate them from the cells, and build a preparation where the biopolymers and the molecular motors are isolated so we can then quantify their behaviors. So this is this simplistic system that just combines these two proteins, the actin biopolymers and the myosin motors in white or in blue. And the myosin motors, when they're walking along the actin filaments, over time start to coalesce and compact all the actin filaments together. So just these two proteins in a disordered system can spontaneously drive the contractility. And so by analyzing this data and doing many different types of experiments, we can come up with a simple theory that I just want to give you the essence of why you can get spontaneous contraction. And the idea is this, is that if you have a system that has is perfectly symmetric and is where all the velocity is the same, you're not going to build any stress buildup. But if you have any small variation in velocities of your motor proteins, you're going to build up internal stresses within the bundle and in the network. Some of these stresses are going to make the filaments under tension, and some of these stresses are going to be under compression. And now, just like you would imagine for a rope can support tensile stresses, very high tensile stresses, but very weak stresses, compression stresses due to Euler buckling. All the regions that are under compression are just going to buckle out of the way. And that sort of provides a symmetry breaking event that only results with only tensile stresses that then drives contraction. So then what happens in these disordered systems is a length scale emerges that is determined by the average distance between these buckles within the system that determines your effective contractile unit. And then we can go in with our microscopy and actually visualize these contraction and buckling events inside while the system is contracting to show that that's the mechanism. Now, what I really like to think about is that these are the same two biological molecules, right? But they build machines with very different types of properties. And so I'm just going to give you a summary of what the differences are between the contractile machines you can build with a sarcomere, or that's involuntary muscle, versus one of these self-organized units, right? That sarcomere is highly dependent on its geometry, and that means it's very sensitive to any amounts of disorder in your system. It also means that unless you really change the structure or the biochemistry of the system, you cannot change the rates of contraction or how much the contraction occurs. You're always limited to about 30% decreases in 30% contraction. However, in our self-organized contractility in disordered systems, it's very robust and insensitive to any disorder. And it's only driven by this mechanical instability. And because it's always sort of a self-correcting process at any time step, it's able to contract very large extents. And by changing the local mechanics, you can really tune the contractile rates. And in fact, since finding these first changes, we've been able to now explore a whole phase space of different types of mechanics that you can get in this motor filament systems. You can actually even make them extensile. You can make a fluid that has a pressure that actually pushes outward rather than contractile. So just with the same molecules, just by how you assemble them into different kinds of materials, you can make machines with very different physical properties. And I think this is one of the most amazing features of biology, that you have these molecular features that can be tuned in very different ways. Now, for the last few minutes, I just want to go through a whole different section of my lab. You know, part of what we do is not only just reconstitute the process outside the cell, but we also take movies of cells. I don't know why my computer's choosing this moment to freeze. Wow, my computer really, if I said this never happens to me, that you wouldn't believe me. In 2 years of a pandemic, this hasn't happened. So part of what my lab does is also visualize fluorescent proteins inside cells and watch and measure their forces simultaneously. So those two movies I showed you were ways of visualizing those protein dynamics. There you go. Okay. Inside the cells, and also measuring the boundary stresses at the same time. Now, the living systems are dynamic, and one of the real problems, as Vincenzo said, the mechanics are constantly adapting. So a big challenge has been how to take these dynamic systems and really have control over their adaptive properties. So we wanted a way to be able to manipulate when forces are applied within the living system without perturbing it too directly. And this has really benefited. This has been something that's new in the group that we've developed over the past 5 years. It's a tool that now we use genetically encodable proteins, but now the proteins, instead of acting like a little light bulb, light controls their activity or localization. So this is a broad field of optogenetics that actually can control neuronal control and neuroscience. In here, inside the cells, what we're doing is controlling the localization of proteins and their function and activity in cells. So here, I'm not going to go through the details of how it's working, but here we have a single cell that's spread on a microscope. We shine a laser light here, and then the increase in intensity that you see here, the brightness you see here, is the localization of a different protein that we're recruiting to that area. And we're recruiting a protein that generates a local force in the cell to be able to apply a little, like a little tweezer within the cell to pinch it and pull it when we want to, with a certain amount of magnitude and a certain duration. So these are the different types of experiments we've done with this, where we shine light and sort of force cells to do what we want. The left-hand movie is one where we're measuring the forces a cell is generating, then we shine a laser and then it generates a little contractile force, and we change the shape over which we activate, and the amount of force that it generates increases depending on if we increase the area. In the middle one, we're activating a little contractile force in the center of the cell, and the flows inside the cell start changing direction. We take out the force and they release. And on the right, what happens is we're applying a force at the interface between two cells that you'd see in a developing tissue, and that force is squeezing that little junction and interface and making that interface change shape. Right, so this allows us in this very dynamic process to have local control that we weren't able to have before. And I just want to give you one result from this of how powerful it is to be able to do this. You know, so we can squeeze at these interfaces, and we can squeeze for a little bit of time, the junctions shorten, and then you take off the light and the junctions recover back to the original length. So the junction, this interface is behaving like an elastic element. It's really boring. It's like a rubber band. But what we can do is increase the duration of our light pulse and ask, does the same thing happen if we increase the duration of the time? And so now in this next movie, I'm squeezing the light pulse for 4 times longer. And what we see is we get a qualitatively different response. Now the junction shortens for a while. It remodels for a little bit longer. We take off the light pulse, and now the junction is permanently shortened, this little interface. And what that means is that the cell is adapted to this external force and modified its internal mechanics. So this approach now allows us to map out the adaptation of the cell mechanics in response to our external probe. It's just like measure any forced response curve, but this allows us to do us in soft living cells. And so we can map out this forced response of the adaptation as a function of activation time. We see a very narrow, rapid transition. And then we can now develop models to understand the adaptation. And the key thing here, I don't want to go through the details, is all this adaptation seems to arise. The rules that the cell is playing for is by sharp thresholds in the adaptive response. So nothing happens at the low magnitudes. You exceed a threshold of deformation, and all of a sudden a remodeling process happens. So there's this threshold in the force sensitivities that we can then reveal through doing these mechanical measurements. And I think there's something important about this in terms of design. So the fact that, you know, I just want to, the fact that we apply a pulse, an adaptive process happens. What happens then is that the whole response saturates, and you can only get a very small distortion. However, if we now take that same activation pulse and now allow it to be separate with a certain amount of rest time in between, we can now find that we can overcome that saturation and response and allow it to go through 2 cycles. And this allows to actually adapt 2 times with the same amount of input signal than if we had one single long duration. And why do I think this is important? Because it's telling you the cell is reading out that not only sort of the amplitude of your signal, but it's also, or the duration of the signal, but it's also telling you how those signals are parsed in time. And this is giving a way for the cells to get information about time-dependent stress fluctuations in its cues that allow it to respond and remodel over much longer timescales than you would imagine from the single macromolecular machines. And I think this is really important. If we think about cells, they're materials that have all these internal stresses. And you think about taking a sheet of rubber, you stretch a sheet of rubber and it will inevitably break if you stretch it too much. Cells are constantly having these stress fluctuations, yet we never see cells rip themselves apart. So there are these mechanisms at play in here to really sort of temper all these fluctuations and allow it to still evolve slowly over time. Okay, so so I want to end with this sort of forward-looking slide. You know, if we think of like, if we've discovered the laws of physics, most of our ways of designing physical machines is taking those laws of physics and then optimizing and forward engineering the design of a car or whatever machine you would like. Now, what we're doing with biology is taking these living materials and then discovering new ways of physics works in these biological matter. And by discovering these new physical laws, we think that we're going to be able to allow us to engineer new types of systems that are based on the design of living systems. Say things that would allow us to do new types of tissue engineering or new types of soft robotics. Now, with that, I'm going to give it back to Vincenzo. This would also be a great time for questions. And thank you. How do I get this?
Any questions to Margaret? Looks like we can go back to Vincenzo now, and then we can ask anybody if anybody has questions or comments. Go ahead.
Okay. I trust you can see my screen. And so one of the things that I imagine you might have taken from Margaret's presentation that I find both fascinating and intimidating is the sheer complexity of the machinery of life. And some of the questions that we're asking, like when we look at this cell crawling, it's essentially how does it move? And you would think that, okay, that entails figuring out the forces that are acting on this cell. But the complexity derives from the fact that, as Margaret explained, the basic ingredient that control that are distribution of multiple proteins, protein assemblies, things that happens at multiple scales. Do not have a simple way to extract a law that tells us what the force is acting on this moving cell as a function of all of this parameter. We don't even know what is important and what is not important. So what we've been doing recently with some success is to try to complement physics-based reasoning or trying to understand the origin of these forces with machine learning approaches that combine the physics and biochemical complexity to produce answer in science. We want, in other words, to construct a mathematical model using not only our own intelligence, but also artificial intelligence. And I would like to give you an introduction of what that means. So here is a very brief introduction to the subject of machine learning and neural networks. I would like to start with a question which is not probably the most complex question you heard in biology. Is this thing that you see on the screen a cat or a dog? Because I know you were thinking, going to listen to this guy answering this question for the next 10 minutes. This is not particularly consequential for the future of humanities. But this question is not as easy if you want to, as we want, if you want to train a computer to provide the answer. And here you're going to see immediately the difference from what I was talking at the beginning. At the beginning, I start with equation. I have an intuition of what's important. I build a mathematical model. Here, I want to train a machine to give me the answer, and eventually even to give me a mathematical model. So let's start with this cat and dog, because it's not as simple as it seems. The basic idea of machine learning, and in particular neural networks, is that instead of taking a piece of paper and writing down a bunch of equations just with your brain, you want to construct a synthetic brain that is capable of providing you the answer. How is that done? Well, for example, in this case of the cat, you could imagine taking a grayscale picture of this cat, making it into many pixels, and then feeding the intensity of this pixel to many artificial objects that we're going to call neurons, that are going to perform some mathematical operations. And then these neurons are going to be connected to other layers of neurons, each of which iteratively will perform a mathematical operation. And by training, that is to say, by updating constantly the weight that connects these neurons, we can get this artificial machine to act as a brain that with a high probability, here 0.9, can tell you that this is a cat and not a dog. I don't expect all the details of the procedure to be clear, but one thing should be clear. At the end of this, we do not have an equation that tells me with 3 terms in it, or 5 terms, or 10 terms, that tells me this is a cat, this is a dog. We have a black box that miraculously gives us answer. And of course, this you can apply to whatever you want. You could apply it to find patterns in the stock market and more consequential and important questions. What we will do it now is to apply it to some of the very complex biochemical questions that Margaret has posed. But the logic will be similar to this one of the cat and the dog. And at the end, we will be able to look inside this black box and tell and convince that machine learning black box to tell us what are the underlying equations that it's guessing using this complicated machinery that mimics our own brains. Okay, so let's go back to a real biological question that's difficult, the one that Margaret posed. So if you want to understand how this cell is going to move on a substrate, well, the first thing you've got to understand is that this cell wants to adhere to the substrate. Now, the typical physicist view is that the adhesion site is like a little spring in which the cell wants to stick to the substrate. Now, when you talk to a chemist or a biochemist, they start laughing because they don't see just a little spring. They see a very complex focal adhesion site where multiple proteins are involved in getting that cell stuck. And guess what? Our problem is that we do not know which of these many proteins is actually responsible and in what manner in creating the forces that get the cell stuck. And if we can't figure that out, we do not know how that cell is going to move. So the plan that I'm going to sketch for you that we've been pursuing with Margaret can be articulated in the following step. First, we're going to use neural network and machine learning to understand which of these many proteins or what pairs of these many proteins is actually responsible. And we're going to figure out that actually we do not require all of the protein to write down an effective model. So first, we're going to identify the proteins that are important. And second, we're going to use machine learning and artificial intelligence to build chemophysical models, using a path that is alternative to bread and butter mathematical building, mathematical model building. So in practice, we're going to proceed in a very similar manner to what we were doing in the example of the cat and the dogs. What we're going to input, however, to these neural networks, instead of a picture of a cat, are fluorescent images like the one that Margaret can take in her lab. In fact, this is right from her lab, of proteins. And there are multiple proteins. We're going to train our neural network showing many fluorescent images of these proteins. Simultaneously, Margaret can also measure in experiment, as indicated here, the forces that exist in the cell. And by showing many images of the fluorescent images of protein and many experimental picture of forces, we're going to train our neural network in such a way that it can produce an answer for what the forces should be. And what you see here on the right is the neural network outputting this picture of forces. This is the actual one in the experiment. And I invite you to see how strikingly similar the two are. Of course, we can measure the errors and are extremely low. Now, the bottom line is that this neural network has also learned that out of all the proteins that are at play, and we know that they're important biochemically, it's enough to give only one, only the distribution of one called Zixen to achieve this type of accuracy. To us, this was very big news. We don't have a model, but we've been able to make an inference on a very complex problem. We know that if we want to know the forces in a cell, the only thing that Margaret has to measure is the fluorescent image, the density of one single protein. And then this trained black box, this neural network, can give the answers that we want with remarkable accuracy. And to bring this point home even more, of course, here we did the same exercise with a bunch of other proteins called actin, myosin, paxillin, zixin. And again, you can see the forces measured and the forces predicted by the machine learning. And the agreement, even just by glancing qualitatively, is utterly striking. There's no other pipeline that can produce this using a simple mathematical modeling approach. And as I advertised before, if you look at these histograms, there is one protein, zixin, here in green, that has particularly low errors. And on the y-axis is error. And the thing that was most striking when Margaret and I looked at this data is that even when we use zixin plus another protein, this is the brown data, we couldn't do much better than just zixin. Zixin had enough information to produce this very beautiful agreement. At that point, Margaret and I asked the following question. At the prediction of this black box, that seems so reliable, also robust. In other words, what happens if Margaret takes one of her cells and does a very violent chemical perturbation, like tries to disrupt the cell by knocking off this protein called myosin that is responsible for its structural integrity. And what I'm going to show you here on the right, it's a graph which I find very striking. On the y-axis, there is the total force in the cell, average over time. And you're going to see the gray data is an experiment where Margaret measures this thing. And the blue data is machine learning predictions. And at this point, labeled by green, a very violent chemical perturbation is applied by Margaret to the cells. And as you can see, the two curves are still on top of each other. The machine learning can predict on a cell that it has never seen before what's the correct force distribution, even as the cell is undergoing very strong remodeling. So now we have a black box that is remarkably good at making prediction. These predictions are robust. The thing that is very unsatisfying from the perspective of classical mathematical modeling is that we have no clue how this black box actually works. We don't know what's in there. We just know it works. And this, while it's satisfying from solving practical problems, is not very satisfying from a scientific perspective. So what I would like to document now in brief is the very recent effort that we've done in extracting from the black box mathematical models. How do we do that? Well, that has got to combine both artificial intelligence and the human intelligence that we've been pursuing in many years of studies of physics and chemistry. So what we did, we said, okay, let's start from the vanilla model on how you predict forces in cell. You start like with a potential energy, a bit like the energy of a spring. But this complicated formulas has three pieces in it. One piece here, this complicated equation, tells you the bulk contraction of a cell. The second piece tells you how the cell adheres to the substrate. It's the one I've been talking about a lot. And the last one tells you what happens, how the edge of the cell contracts. In the past, people had tried to use this mathematical formula to find the forces on a cell. Using this continuum model, they produced pictures like this. This is actually from previous work that Margaret and some collaborators have done. Now, I invite you to compare the picture called continuum model with the picture called experiment. I think even from a casual glance, you can see that the continuum model just predicts forces at the edges, in any way, the heterogeneous distribution of these forces, of course. Whereas on the right-hand side, you can see our machine learning algorithms can do a wonderful job at capturing this inhomogeneity, spatial inhomogeneity. Why? Because they know how the proteins are distributed. So the question that we had is, can we combine something about this model that seems to work with information from machine learning and construct an analytical model like this, which is informed by machine learning and superior to the one that you construct just with your own brain? So what we figured out first is that actually of these three parts of this equation, one of them is not even necessary. And we learned that by adding data extract from machine learning. We learned that this edge contraction, the contractility, is not important. We can just get away with the first two terms of the equation, but we need to enhance them. And we need to enhance them in a way that we learn from the data that Margaret has collected using artificial intelligence. What did we learn? We learned the following. This coefficient called y that tells you how the cell adheres to the substrate is not just a constant, like our simple mathematical model we're predicting. It is actually a function of space. So we have to modify our mathematical model in that way. But you may say, well, look, okay, sure, it's a function of space, but who tells you that function? Well, the way this thing adheres to the substrate depends on how many of those Dixon proteins are there, something that we discovered using machine learning. And machine learning also can tell us the exact dependence of this y coefficient on the density of the protein that Margaret can measure. Once we combine these approaches, we have a model that is simpler than the previous one, but is informed by neural network in promoting two terms to be dependent on the density of a chemical. Once we do that, I want to show you what we can do by combining this analytical equation with a neural network of a more complex type than the one that distinguishes between cats and dogs. And this is shown on the next slide. So what we've done, we've taken an analytical model, we inputted information about the density of proteins from machine learning, the shape of the cells, and then we can solve it exactly because it's an equation after all. And we have produced what we call a continuum mathematical model plus machine learning. So the starting point that we had was nature, which is this picture of the forces recorded by Margaret in the experiment. Deep neural networks, the black box, can do a wonderful job at capturing it. And the continuum model done by our brains wasn't as good. Now, in the middle between these two, you'll see a fourth image appearing. This is the continuum model plus plus. This is still an equation, but it still has been guessed by the neural network. As you can see, is vastly improved compared to the continuum model that we've just built previously using physical principles. In particular, notice that it captures the non-homogeneous distribution of forces at the edge of the cell in a way that matches not only the neural network, but also the experiments. Okay. Now, the main advantage of this continuum model plus plus is the following. If you look at nature, the experiment, versus the deep neural network, the deep neural network is still the best that we can do. But the continuum model plus plus is something that we can understand. So if you like, an auto-mathematical modeling that emerges here is that you could almost have a plot of descriptiveness versus complexity. Now, the thing that we do with deep neural networks, it's very descriptive. It does a wonderful job at capturing reality. It can even tell us information of what proteins are important, but it doesn't tell you much about the basic mechanism underlying it. It's low on physical insight. On the other end of the extreme, there are mathematical models that we build out of physical principle. They have a lot of insight in it because we start with physical insight, but they don't do very well in describing reality. The last thing that I showed you, which is in the middle, this continuum model plus machine learning, is an attempt to combine the power of prediction of machine learning and data-intensive technique, artificial intelligence, with our own intelligence to construct models that are predictive and interpretable. Now, with that, let me conclude. So what we started with a classic approach to building machines. You start from physical laws, you modify them as you want in exotic ways, and you can build complex functionalities, like a wheel that goes uphill in an adaptive way. But what we're confronting with functioning when studying biological machinery is something that requires a combined approach that combines the physics understanding that we have of the fourth generation with machine learned approaches. And our goal in the next few years is to apply the pipeline that you've seen to many other problems in which we can use neural network to actually produce mathematical models that are interpretable and that will complement our own intelligence with artificial intelligence. Thank you very much for your attention.
Thank you, Vincenzo and Margaret. There are excellent presentations with outstanding work and amazing results and very fascinating. I'm certainly very excited what you have been doing. I don't see any hands or any questions in the chat box. So let me start with the one question. Your research work seems to me that you'll get a lot of benefit by collaborating across different disciplines. This is beyond the physics you mentioned about chemistry and also biology, mathematics, and computer science and all that. Again, this is a question to both Margaret and Vincenzo. In this regard and beyond, is UChicago unique for this type of research? And also is now the current time something special?
Let's see, I'll speak for myself. So I think what makes UChicago a really special place to do this is how compact our campus is. Right, so I've been collaborating with biology colleagues across the Green and Cummings for 15 years, and our labs have had joint students over that time. We also have a really unique biophysics graduate program, which is really the only one of its kind where students come in not it's not a disciplinary program. It really allows students to choose whichever advisor. So it really allows us to keep evolving what interdisciplinary boundaries are hit from the biological and physical sciences. And I think that's really exciting. And then, of course, you know, both Vincenzo and I are in the James Franck Institute, and this is a really special culture where there are a lot of a strong culture of postdocs that are and joint mentoring between different faculty. And so I think there is a really nice culture of collaboration that is occurring here, but definitely in the areas of sort of active matter. Chicago has been really driving this forward over the past 10 years. So I think it's pretty unique in that regard. Vincenzo might want to add something.
Yeah, I echo Margaret on this. This type of studies not rely on one thing only.
Yeah.
They rely on knowledge of mathematics, knowledge of computer science, knowledge of chemistry, knowledge of biology, experimental and theoretical techniques. Having an institute like the James Franck Institute, where Margaret and I are, where a group of people with all of this background sits virtually on the same corridor, is where this type of things can happen.
Yeah.
And
buildings like the Gordon Center, right? I mean, so my lab is my lab is split in the Gordon Center where we sit, right? There's biological sciences on their side of it, and that's been really essential for my group too.
Great.
Yeah.
As we say, you know, this interdisciplinary research is in our DNA. So
right.
This is great. So there's one question from the audience. Thomas Zimmerman asked, how do cells communicate attractions or stresses to each other?
Yeah. So there are there's another there are another cell proteins, transmembrane proteins that connect cells to each other, including things like the cadherins proteins that form the same types of specific interactions that cells go to the outside world. So they pull on each other, and there's also signaling hubs there that stimulate growth factor signaling and cell proliferation of whether cells should go to the cell cycle. There's other junctions too that allow ions to pass through cells. So yeah, so when you have multicellular tissue, there's specialized connections between cells that allow for transmission of different kinds of signals, which is actually something that Vincenzo and I were talking about an hour ago, about work that we're doing in that in that regard.
Great. Any other questions from the audience? You can raise your hand physically or using reactions button or chat box, any way you like. Maybe I'll ask another question. Can you think of any applications from your research results? What could it be other research areas, or could it be, you know, more society or industry, whatever that means?
Do you want to go first, Vincenzo? Or
yeah, certainly for robotic aspects of it, it's very engineering driven. The idea of you know, currently you think of a single robot that does something, but crucial in all of the things that we're thinking about is what happens when you have an ensemble of robots, a flotilla of it. Can you start describing as if it was a material? How would the 100 robots or 100 micro robots perform a task? And this is a question of intense technological interest. Our approaches, which are also inspired by how nature behaves, has something to say about that.
Yeah, from our perspective, I mean, I think that so I like to think about the design principles of that biomolecules use, right? And so we can take the route of making what is the minimal components that we can make with synthetic polymers to get the same type of response, right? We're not going to be able to re-engineer a protein. Proteins are only stable; they can't be deployed under lots of conditions. But can we identify new design principles? Such as this buckling phenomena, right? And that we can sometimes use those to build new types of soft matter. But I'm also finding more recently that just the framework, the development of a sort of biophysical framework of how to characterize force transmission has allowed us to start making inroads into understanding just basic biology questions of cell proliferation and cell size regulation that had been tissue biology that had not been done. If we think of all the synthetic biology that's been done in the gene engineering, it's all at the single cell scale without any mechanics involved, right? So this has all been done through gene regulation, but we know that mechanical forces are really essential for controlling gene regulation and multicellular, and it always happens in the context of multicellular tissue. And so I'm seeing now that these ideas can be really helping us understand regulation of expression patterns and sort of cell differentiation and cell fate specification in multicellular tissue, which I'm really excited going forward.
Great. I'll have one more last quick question. Thinking about future technical developments and both hardware or software, including machine learning, but what are the future sort of developments that you can imagine or you can dream of?
So, I mean, right now, I mean, the future for me is still all the work we're doing with Vincenzo. None of that is published yet. So I think we've been at a block of really trying to figure out how to capture, identify what length scales in active systems and living systems are important for regulating the adapted matter. And I feel like we're at the cusp of being able to make inroads into that. So that's something I'm very excited about right now.
Great. Actually, there are one more
questions
from the audience. So let me say, Francisco Faraco asked, could Vincenzo comment very briefly on machine learning or AL project that he has done in the connection with the stock markets?
Sorry, you can probably hear me now. The thing that we're doing that is closest to that application is that in the stock market, often you have to analyze time series. And the thing that is extremely complicated is the huge amount of noise. This is a problem that exists also in actually the question related to the previous question, how cell-cell interactions happen. It is also dominated by that. So one thing that my group has been doing, which is not analysis of the stock market per se, but time series that type of structure is ECG patterns that come from recording signal from the brain. In particular, with people here, we study epileptic brain and ECG patterns that come from it. And we try to look for clues in those very noisy time series for epileptic event that will happen. Now, this problem is very close from a data analysis perspective to understanding, if you look at a time series of an index, to try to figure out when a crash in the stock market will happen in the future. It's a problem of an incredible complexity. A similar thing happens for signals for earthquakes. And the problem is the extremely noisy nature of the data. And with Margaret, we've been using a technique called graph neural networks that is particularly useful to predict this type of time series where huge amount of noise are present. We haven't directly applied it to the stock market, and if that worked, it would be great, but we have applied it to extremely noisy data. And this is, of course, a very hot area of research.
Yeah, I can imagine. Okay, with that, I would like to thank all of you to join today's conversation and discussion and incredible presentations by Margaret and Vincenzo. I'd like to thank all of you. Special thanks goes to Sean and Amy to put together to make this beautiful and wonderful event. So thanks to all of you.
Thank you. Thanks.